Hi everyone, so today we're going to discuss what is cancer and how it is different from regular cells. So first of all, cancer cells are physically different from normal cells. Where normal cells are uniform in cell size and arranged into discrete tissues, cancer cells have variation in cell size and shape with a disorganized arrangement of cells. And cancer cells also have large variable-shaped nuclei with coarse chromatin and a small amount of cytoplasm, which differs from normal cells' small uniform nuclei with fine chromatin and a large amount of cytoplasm. Cancer cells can even have multiple nuclei sometimes, and cancer cells divide a lot more than normal cells and do not demarcate cell tissues or functions, and they have poorly defined boundaries and lose their specialized features. And so then we move on to tumors, which are a collection of cells, and they can either be benign or malignant. Benign tumors grow locally and stay local and are non-cancerous, while malignant tumors are cancerous and can invade neighboring tissues and spread to other parts of the body through blood vessels. And so there are four main types of cancer are carcinoma, sarcoma, leukemia, and lymphoma. Carcinoma grows in epithelial tissue in and outside of the body, such as skin or organ linings. Sarcoma is in connective tissues in bones, tendons, cartilage, muscle, and fat. Leukemia is cancer in the blood that begins in bone marrow. Finally, lymphoma is in the lymph system. And so, on average, 6 to 10 genes need to be mutated for a cell to become cancerous. These mutations could be either somatic or germline. And somatic mutations are mutations in body cells, while germline mutations are in the egg and sperm cells, passed on to offspring. So cancer running in families may be influenced by these mutations. So that's the difference between inherited and sporadic cancer. Inherited cancer is caused by an inherited germline mutation and then additional somatic mutations that occur to actually turn the cells cancerous. And sporadic cancer is solely caused by the accumulation of somatic mutations throughout life. And so how do somatic mutations occur? They're mostly caused by mistakes in DNA replication that have not been caught by the checkpoints in the process of replication. So when replicating, cells might make a mistake and mismatch a pair of nitrogenous bases, and it might not be caught in the checkpoints, or the checkpoints are not even functioning because of the mutated tumor suppressor genes. Then, when this strand of DNA replicates, it creates one copy that's the same as the original, but one completely mutated copy that had a different set of bases in that place, and that can potentially make it cancerous. Environmental factors can also lead to the cause of somatic mutation, and so some of these environmental factors are smoking, which is the biggest one, and it can cause different types of cancer all over the body, specifically in the lungs, mouth, throat, and esophagus. And being overweight and a poor diet are also the next biggest factors, because fat cells produce more hormone and growth factors, which causes more cell division, which increases the chance for mistakes in replication and creating cancer cells. A good diet reduces chronic inflammation. Antioxidants with a good diet neutralize DNA damaging free radicals, and it promotes immune system function to fight off any harmful cells. And drinking also increases the risk because when a large amount is drunk, there's a buildup of acetaldehyde from all the ethanol, and acetaldehyde can cause DNA damage, chromosome arrangement, and for DNA to bind and form clumps, increasing chances for somatic mutation. And drinking 1 bottle of wine per week is the same as men smoking 5 cigarettes and women smoking 10 cigarettes. And so then there's the two-hit hypothesis that says that both copies of a gene, because there's one copy from each parent, need to be mutated to turn off a gene and get you closer to developing cancer. Those with germline mutations are often even closer to developing cancer than others, because while it's not guaranteed, they only have to develop one somatic mutation in that gene compared to the others who need 2. And proto-oncogenes are genes that stimulate the cell cycle and trigger cell division by producing growth factors, hormones, and other proteins to carry messages. So when they get mutated, oncogenes are produced, and they accelerate the cell cycle. These mutations cause a lot more cell division than normal, which allows for cancer cells to reproduce really quickly. And so this is a hallmark of cancer called sustained proliferation. This is when cells have mutations in their proto-oncogenes that overstimulate them. Many cancer cells can synthesize and secrete their own growth factors, triggering them in neighboring cells to divide. They can also have more growth factor receptors, making them more sensitive to cell division triggers. And other mutations can also lead to the overproduction of cyclin, which allows cells to go through checkpoints. Other genes that may get mutated are tumor suppressor genes, which regulate the cell cycle to protect cells from developing cancer. They look for mutations, and when tumor suppressor genes get mutated, cells can keep dividing even when they have really harmful mutations that make them cancerous. The combination of muted proto-oncogenes and tumor suppressor genes leads to the development of cancer because cancer cells can reproduce at a quick rate without being stopped to fix their mutations. Which, another hallmark of cancer is the evasion of growth suppressor signals. This is when they have mutations in their tumor suppressor genes that inactivate them so they can go through checkpoints with DNA damage. Resisting cell death is another hallmark because when normal cells have DNA damage or their telomeres are shortened, they initiate apoptosis or cell death. And cancer cells with mutated tumor suppressor genes and other cell damage will not receive the apoptosis signal, allowing them to continue to divide and grow tumors after the point when normal cells would die. And another hallmark of cancer is replicative immortality. So normal cells have telomeres on the end of their chromosomes that protect the coding part of their chromosomes, since a chromosome gets shorted every time the DNA is replicated. But cancer cells have the enzyme telomerase, which lengthens telomeres and lets cells continuously divide pass on their damaged DNA to daughter cells. Cancer cells can also have an abnormal number of chromosomes in addition to mutated tumor suppressor genes and proto-oncogenes. So they may have 3 of some chromosomes of different lengths, or missing chromosomes, and this could be a problem because extra proteins may be synthesized or some may not be made at all, meaning that cancer cells lose their specialized functions they had as normal cells. And while normal cells carry out aerobic respiration to produce ATP and glucose, cancer cells use aerobic glycolysis to rush through interphase. It produces less ATP, but it works faster, and therefore cancer cells consume a lot of glucose to do this. It also produces lactic acid that cancer cells can use to turn into materials to produce new cells, which carbon dioxide cannot be used for. So normally T cells and macrophages work together in the immune system to get rid of the unwanted foreign cells that are there. After that process is complete, they bind together so that the macrophage can tell the T cell that the job is done. However, some cancer cells can have the molecules on their surface to shut down T cells, even when the malignant cancer cell is still spreading. So this is another hallmark of cancer, which allows them to keep spreading without being shut down. Inflammation is localized swelling in response to injury or infection. And when this happens, blood vessels bring more white blood cells, which are growth factors, to the area to deal with the inflammation, because they want the cells there, like if you get a paper cut, they want them to replicate to cover it up. So inflammation can be temporary or chronic, and chronic examples are the asthma or cancer. And so this creates a cycle of inflammation, bringing growth factors, which then leads to the division of cancer cells growing and spreading, which causes more inflammation. So this vicious cycle will promote the growth of tumors and cause this hallmark of cancer. And angiogenesis is the development of new blood vessels. It is involved with cancer cells when they release a chemical called vascular endothelial growth factor, which makes blood vessels grow toward the cancer cells. These newly formed vessels can be leaky, which allows for cancer cells to get into the bloodstream. And when cancer cells get into the bloodstream, they can metastasize, which is when cancer cells spread from their original location to another part of the body. Cancer cells are not connected through the extracellular matrix and lose their adhesion, which allows them to enter the bloodstream. They can then land in a new location and create a new tumor. This is why it's important to catch cancer at an early stage, so it can be treated before it spreads throughout the body, and it's hard to find all those loose cancer cells. And so pedigrees are tools that are used to track certain trends through a family and to help find germline mutations. And so members with a certain type of cancer are marked, and when there's a trend that matches with DNA testing, later generations know to be wary of their health and be aware of certain environmental factors to try and prevent getting cancer. And that's it. Thank you for watching.